Welcome to the Boardwalk Talks program brought to you by the Alabama Aquarium here at the Dauphin Island Sea Lab. My name is Mendel Graber. I am one of the educators here at the lab, and today we are over in one of our research labs and we'll be chatting with Randy Cannon, a grad student here, and we're going to chat with Randy about phytoplankton and about some of her work, like kind of doing algae gardening.
Yeah, that's a perfect word for it, actually.
So with that, I'll let Randy tell you a little bit about it.
All right, cool. So our lab works, like Mendel said, with phytoplankton. And phytoplankton are these cool little guys that are found in our oceans. They are tiny plants, essentially. If you took out a cup of water and went down to the beach and scooped up some water, you would find thousands of them in that water. You couldn't see them with the naked eye, but if you put them under a scope, you'd see all these little guys. And so in our lab, we like to work with these critters and study them and find out essentially what makes them tick. But the hard part is when you're studying them, you can't just go down to the ocean and take some water and then work on them. You need essentially your culture to actually mess with them and do an experiment, because if there are other things in the water, that's going to mess up your experiment. And so a lot of the work that we do in the lab requires us to maintain pure cultures of these phytoplankton. And for my work, I focus mainly on diatoms, which you can see in this picture. Diatoms are made of silica, and they're just another type of phytoplankton. Thank you. And so for my work, I need to culture these guys, keep them happy, keep them alive, so that I can get solid results that are actually feasible and reproducible. And just like any living animal or plant, phytoplankton and diatoms require a couple things to be happy, and they are photosynthesizers. So we have to provide them with light, and along with that light, they need nutrients, which is essentially a food source. And so in our lab, we have these really big fridges, and they're actually incubators. And so we can set a specific temperature for the interior of the incubator, and these diatoms, bacteria, whatever are in there, will live at that specific temperature, and we also provide them with light. And so in this incubator specifically, it's a 12-hour cycle. So for 12 hours, they get light, and for 12 hours, they get darkness. So we're simulating the same environment as you would find in the ocean. And this is what keeps these guys happy. Alongside that light, though, you gotta have nutrients. And so every time we make a culture, we have to essentially make media, which means I do go down to the dock, I collect fresh seawater, but I can't just put those diatoms in that fresh seawater, because like I said, you can't have any kind of invader in the culture. So I have to filter that seawater through a 0.2 micron mesh, which is teeny tiny little holes. It basically looks like a piece of fabric. Filter out anything that might be in that seawater, and then I take it and I add the nutrients, things like sulfate, nitrates, phosphates, metals, and other random nutrients. We add that to the water, cook it at a really high temperature just to get any other bacteria out of there, and then I can put my culture into that media and grow tiny plants. But you can't just leave them there. For those of you that are plant fans, you know that when you are growing a plant, they kind of outgrow their pot at a certain point. Those roots get too big. And so the same thing happens with my diatoms. If the culture gets too big, I have to split it up or move it into new media, because they will run out of nutrients. And these diatoms can live for a while without those nutrients, but they will supplement that through either growing slowly or not reproducing as well, and I don't want that to happen. I want these to be happy diatoms so that my experiments go well. And so when I worry about them running out of nutrients, I will either move a culture into a fresh batch of media, or I'll take a little bit of those diatoms out and just start a new culture all over again. And it's pretty simple, because these guys reproduce just by splitting in half. So I don't have to worry about them finding a mate or finding a partner and reproducing that way. The plants will literally just cut themselves in half. And what you're looking for when you're culturing is about a division a day. So if I start off with about 300 diatoms per milliliter, the next day I hope to see about 600 diatoms per milliliter, because they're cutting themselves in half and therefore doubling. So the question comes about is, how do I monitor that growth? And this is actually my least favorite part of culturing, because to monitor growth, I have to take out a sample of my culture and put it on this slide. And I don't know if you can see, but there are tiny, tiny little grid lines on this slide. And there are 1,000 tiny squares on this slide. And so I will put my culture, or a milliliter of my culture, into this slide, put it under the microscope, and then I will randomly count how many diatoms are in 50 of those squares. And I just have to pick 50 random squares. And that'll give me an estimate of how many diatoms I have per milliliter of sample. And so it takes me about 30 minutes to count 50 of these squares on the microscope. It's the most time-taking part of my job. Definitely my least favorite bit. And actually, you can see, I don't know how well you can see it, but that's about what the square might look like under the microscope. And these tiny, tiny little dots are my diatoms. So that's what they look like under the microscope. They still look tiny, and that's why you can't see them with the naked eye.
How often do you do that counting?
I shoot for about every other week, just so that I can really monitor. If I'm just doing culture maintenance, I want to monitor how that growth rate is going.
You do it every day that week, or just one day?
Just one day.
And do you just do one slide?
No, it depends on how many cultures I have, right? So if I have 4 bottles of culture, I need to do at least 1 slide per bottle, and I usually like to do 2 just so that I have more of an average, right? Because 1 slide, that milliliter could be a little bit funky. It could have more aggregates in it than another one. So it's better for me to do multiple counts. So it's about an hour every other week, depending on how many bottles I have. It's not the worst thing in the world. But when I have an experiment and I'm actually running trials, I'm taking daily samples of my cultures to see how they're responding to whatever experiment I'm running. And that means daily cell counts. And that's when you truly see how fun science can be and the challenges that you have to deal with to make sure everything's running smoothly.
So what kind of experiments are you doing that require these cultivated algae?
That's a great question. So I'm in 2 labs. I'm in the Physy lab and I'm in the SME lab. And the Physy lab focuses on algae, and the SME lab focuses more on ecology. So I'm trying to intersect the 2 of these ideas. And I'm doing that by growing diatoms, which will go in this little tube. This is a mesh that's so fine you can't really make out the holes. It's a 5 micron mesh. I will grow diatoms in this tube, and then they are surrounded in water that is filled with copepods. And copepods eat diatoms, so this is a natural predator that you would find. And so I'm essentially trying to scare the diatoms into emitting some kind of response when a predator is present. And there's been multiple experiments done that have shown that diatoms emit a response, whether it is increasing the size of their shell or their test. They'll actually kind of bulk up, essentially, to try to be harder to eat. They will form chains. They'll link up with other diatoms and become a long chain, because once again, that makes them harder to eat. Or they'll aggregate, which is a lot like forming a chain, but instead they'll form bundles. And so I'm trying to basically see if I can get them to bulk up and form a thicker shell.
So you mentioned that their shells are made of silica, so it's sort of like glass.
Yep.
So a couple of questions this brings to mind for me. You are kind of sort of examining kind of grazing pressure. Mm-hmm. So if we thought of this as maybe something analogous to, animals grazing on plants, something that might be a little more familiar to people, a little more visible. So some plants benefit from grazing. Is there anything analogous to that with diatoms?
That is a great question. Not that I have seen or read about, but I wouldn't be shocked if there would be a similar situation in which they do. I know that we see the same way that plants on land will emit toxins when they're being eaten, or they'll be toxic and therefore try to fight against being eaten. Diatoms will do the same thing, right? That's why we see blooms that turn the water red, because these phytoplankton are trying to be toxic and fight against being eaten.
And can you point out some of the differences between true plants, vascular plants, and these diatoms?
Yeah, so vascular plants and diatoms both have cell walls, but that's kind of where a lot of the similarities end. They also have, vacuoles and a lot of that similarity on a cellular similarities, but from there we kind of differ in that really this glass-like test is something that we don't see in plants on land. Plants on land don't have a lot of that same kind of structure unless you're looking at a tree, right, which has bark and a solid, firm wall to stop them.
So can you see it in this Tudonicea, what you're talking about?
Yeah, you can see this wall here.
And is this 3 segments?
Yeah, these are 3 segments linked up.
So you started with one and then it separated itself. That's what you talk about when it splits?
Not necessarily here. This is more looking at where they will form chains. I wish I had a picture of them
Petri dish?
splitting. Yeah. But when they split, it's a lot like having themselves, and you can actually look at this here, like one half of this Pseudonicea, if it were to split, it would split down the middle, and so it's essentially creating 2 twin daughters that are exactly the same, and it has everything that they need to be on their own.
So you're measuring a response, but if I'm understanding correctly, your 5 micron mesh. Can we show that again? And just to explain what 5 microns is, that is 5 millionths of a meter. But the copepods they can't physically access the diatoms, is that right?
That's right.
So they must be responding to some kind of chemical signature?
Yeah, some kind of cue. And what we're not sure about is if they're responding to the actual signature of the copepod, and if they can sense something going on there, or are they responding to, because I will feed the copepods on the outside other diatoms, because I have to keep the copepods alive. So the other question is, are they responding to essentially their brethren dying? Are they getting that cue of, oh, I sense a dead diatom, maybe I should bulk up so that I'm not next? And so
an interesting comparison would probably be oysters, correct?
Yeah. And my lab does a lot of work with oysters, and we do the same thing of these oysters bulking themselves up in the presence of predator cues. And there's been some great research on that. They do exactly the same thing.
So when you say they bulk up, what is, you're referring to them kind of thickening that glass test. So are you looking at the mechanism by which, not just like what stimulates that, but also like the abundance of the mineral, or how they access it, or how they pull it out of the water?
Yeah, so they will pull silica out of their surroundings and use that and lay it down in their shell. And so I will essentially dye the water with a luminescent dye, and that when they're taking up the silica, they're also taking up that dye for me. So I can see how much dye there is per diatom, and that tells me how much silica they've taken up.
And how long does this take?
So the last experiment I did, we found that it took about 7 to 10 days to actually see that response. We didn't see a response in the diatoms the first 1 to 5 days. It was a little bit later that we saw them actually responding to the predators present.
Could you explain it in terms of, so with the thickening of that test, so can you tell, is it twice as thick as before it was exposed to the copepods, or is it?
With mine, I cannot tell. I have not been able to really dial in on that just yet. I've only done 2 rounds of the experiment, and the first round, we hit a bit of that, usual scientific bump in the road as it is. And so the last experiment, we actually got some more solid data. So I'm hoping when I perform it this third time around, we can see that a little bit closer and see really how much of a difference there is between it.
And how do you plan to measure that?
So I measure that by comparing the samples of a control, diatoms that have not been put under essentially any stress, haven't had to deal with any copepods. And then I have my diatoms that have been put under that stress and have that cue of the predator. And I take those samples, I filter them so that I'm just looking at the diatoms, and then I will perform a digestion, which means I'm dissolving and getting rid of anything that's not silica. And then I will compare the amount of silica in the control and the experiment. And so that'll really show me, like, the silica is what's supposed to be changing. So that will show me, okay, is there more silica when a predator is present?
I assume you're working with very small quantities. So how do you measure that?
I am working with very small quantities. And so I will measure that with light. I will put them in a tube, put them in a machine, and however much light is refracted through my samples is supposed to tell me how much silica is present.
Why do you start with seawater?
Because that's what these guys are happy with. I could make seawater myself, but it's easy. I mean, it's just as easy to go down to the dock and collect seawater.
And filter it and clean it, and
filtering happens pretty
sterilize
quickly. The filtering happens pretty quickly. It goes through that mesh at a pretty fast rate. And even if I was using like created seawater, if I was making that myself, I would still have to sterilize it. Like, I would still have to autoclave it, cook it at high heat, high pressure, just because that's part of the process. And bacteria can be introduced at any point, which is also something that I'm currently working with. So whether it was created seawater or filtered, the same thing happens.
And where do you get your diatoms that you start with?
So I inherited my diatoms from a fellow lab mate. This culture is obviously not the same because they've been dividing and dying off. So it's a new culture every couple of days. But I got that from my lab mate, Alex. He bought that, I believe, from another lab, Bigelow, I believe. So you can just essentially buy these cultures on the internet. If you wanted to culture at home, if you're that much of a plant enthusiast, you could just buy this online, and the culture is shipped to you.
What species are you working with?
I am working with the Lasiosira rotula, and as their name implies, they're round, rotula, round. They are a chain-forming diatom, but I have not seen them form chains in my cultures. I'm not sure why that is, but as far as us growing them, they're not forming any chains for us.
So if these are sort of lab raised, going back maybe generations, what kind of genetic diversity are you getting?
That's a great question. I'm not sure. I don't do anything on the genetic spectrum, so I'm not positive.
Because that might introduce some kind of, variable to your...
No, that's for sure, because that could be why we don't have, they don't form chains for us, is there's some kind of genetic diversity there, as well as if the diatoms that I have were to survive my experiment, if I didn't, filter all of them and kill them, it could be that there's some kind of genetic passing on of this trait of increased silica in their tests. That would be a cool thing to look at.
So, with this division that diatoms do, if we just talk just a little bit about, reproduction and genetic diversity, how do, if they're just dividing, how do diatoms as a population increase genetic diversity?
Well, that's also a great question. I think that it works a lot on kind of the same frame of what we're looking at, as some kind of stressor is introduced to these diatoms, whether it be predation, temperature of the water, salinity, anything like that. These diatoms are obviously responding. We have lots of evidence of that, many published papers. And so as a response, they're altering their physiology. And so then when they're dividing, like I was saying, they pass on literally all of those traits that they've built within themselves to their daughters because they are of themselves. And so that's, you're looking at genetic diversity in like working right there, right? We could see that on a daily scale. So every division essentially could be passing on new traits within the scope of these these diatoms.
So they're let's go back to what you explained before. They're doubling their population daily.
Yeah, daily if they're happy.
Essentially. And how long do they live? So, like, how many generations are you working with?
So in a culture, if I like were to take it out and look at it, I'm probably looking at about 2 generations, maybe 3, if there's some that are a little bit slower. But yeah, their life is essentially a couple of days, because as soon as they double, that's no longer the same diatom. That's 2 new diatoms. So yeah, it depends on their growth rate, but you're looking at a lifespan of like 1 to 3 days, I would say, which is it's over in the blink of an eye. Which means when I'm performing my experiment, I can't just put copepods in for 1 day and have just 1 generation of diatoms aware of that. I have to make sure the copepods are staying alive and are being a constant source of stress and cue for every generation of diatom. Otherwise, there's a possibility that some of these diatoms aren't aware of it and therefore aren't responding.
So are your diatoms, are they reproducing within that the mesh as well?
Yes. Yeah. The only way I could stop them from reproducing is to essentially not provide nutrients or light.
So how do you separate out the difference between whether you're seeing them respond to some kind of chemical that the copepods are producing versus something that the diatoms that are being eaten are releasing?
That's a great question, and I would love to get into that as soon as I essentially can prove that there is a response. And I think a great way that you could do that is you essentially take the copepods out of the equation, right? And so at that point, finding some way to crush these diatoms myself and then introduce them into the environment. And so if there's no response to just crushed dead diatoms, then it must be that they're responding to the predator cue.
So where are you getting your copepods?
So I get my copepods from GCRL.
GCRL is? Gulf Coast Research Lab.
Thank you. Gulf Coast Research Lab over in Mississippi. They have a great aquaculture center. They grow oysters, fish, and they grow copepods as well as diatoms. And so they're very kind to provide me with those copepods. I just will call them up and be like, hey, I'm performing an experiment. Could I get about 4,000 copepods? And they're always kind of surprised because they're used to people asking for copepods on more of a number of like 20,000 or so. And so anytime I'm like, hey, I just need a couple hundred, they're like, oh, no big deal. And so they will either give me copepod eggs, which I then maintain and grow out into adults, or they'll give me the adult copepods, which mean I can immediately dive into an experiment.
So how come copepods and diatoms are both occur in our water right off the sea lab, or off Dauphin Island and in the Gulf? So why are you using aquaculture as opposed to just collecting out?
That's a great question, and the answer is time. If I was to go out to the water, get a bucket, and then put that under the microscope, because I can't see any of these creatures with my naked eye. Sometimes you can see copepods, but it's a challenge. I put it under the microscope. I would have to pipette out single diatoms and start a culture there. And even if I did that, there's a chance that I picked up a bacteria or some other tiny, tiny little thing that I still can't see. And so, yes, you could do that, but it is a lot of time. And the same goes for copepods. I would have to pick them out of the water one by one.
So let's put this in a broader scope, because you mentioned the ecology of these interactions. So diatoms are part of the phytoplankton community, the plant like the photosynthesizing plankton. And plankton, let's just kind of go back to basics a little bit. Plankton is a lifestyle rather than a taxonomic group.
Yep.
So plankton are those organisms that drift with the currents, and we kind of separate them into the ones that are plant-like, phytoplankton, and then the ones that are animals or animal-like. And so diatoms are phytoplankton, copepods are zooplankton. Among the zooplankton group, some of them are plankton their whole lives. Copepods are plankton their whole lives. And then a lot of organisms in the ocean will begin life as plankton. So if you think of caterpillars, caterpillars turn into butterflies. Think of tadpoles, they go through a metamorphosis, they turn into frogs. So there are a lot of animals in the ocean that go through a metamorphosis and they begin lives as part of the plankton community. There are other photosynthesizing organisms in the ocean, such as seagrasses, right at the boundary between land and sea here in our temperate climate here in our estuary system. We have a lot of salt marsh grasses, but out in the ocean, and there are also macroalgae. So these are what we've been talking about are microscopic, but there are also seaweeds that are essentially they can be seen with the naked eye, and they are algae. So seaweeds are different kinds of algae. So there are other kinds of primary producers, those organisms that can use sunlight and nutrients and produce food, and that's the base of the food chain. So the phytoplankton is a really important component. It's the base of the marine food chain. So if we put that in kind of a broader picture ecologically, the diatoms are an important part of the food chain, and then often it's the organisms that we can see that draw our attention, but these microscopic organisms are vitally important to this food chain. So our attention may be grabbed more by red snapper, but the red snapper are dependent upon these organisms at the base of the food chain. So you've got copepods that help transfer an awful lot of biomass or food that's available, that's phytoplankton, and then something bigger like anchovies or other kinds of zooplankton that are bigger than the copepods might come along and eat the copepods, filter plankton out of the water, and maybe they're a little bit bigger and they can be eaten by animals that are a little bit bigger than that. So in that way, there's an awful lot of food that is phytoplankton. We sometimes call it the soup of the sea.
Yeah.
And a lot of animals can't target it. Mm-hmm. So they have no way of eating from that level, that trophic level, that level of the food chain. So they depend on animals like copepods and then filter feeders like anchovies to transfer that food supply up the food chain.
Yeah.
So what are maybe some of the broader scope implications of this relationship?
Yeah, so looking at the food chain, something that I found is if these diatoms are changing their size based on the predator cue, then some of these diatoms will decrease or make themselves smaller or not form chains. And so by making themselves smaller, it's harder for copepods to find them. And so that leaves them open to being grazed on by protozoans or some other smaller predator. And so by doing that, you're creating another link in that food chain. And we know that about 10% of energy gets transferred every link in the chain, right? So adding another link is decreasing the amount of energy that you're finding, making it to that top level, trophic level. And so really understanding these connections and finding out, because they're the basis of the food chain, finding out how these physiological alterations affect that transfer of energy is kind of an important question.
So when you talk about the transfer of energy, you can compare it to an apple. A whole apple is going to give you a lot of energy.
Yep.
But a piece of an apple, and that's basically what you're looking at, is a larger diatom that goes higher in the food chain offers more energy in the food chain, correct?
Yeah. Yes, yeah. And you're also looking at the level of food provided. So when you are changing your structure, there's a trade-off, right? You can't just study all day and not eat and sleep and feel fine the next day, right? There's going to be some kind of trade-off. You're going to be tired, you're not going to be able to focus. And so same thing happens with animals. So when these diatoms are bulking up, they're putting a lot of that energy into building that test. They're not putting energy into having as high of a nutrient content within themselves because they're not focusing on feeding themselves with nutrients other than silica. And so these copepods are then eating essentially a lower level of food.
Like a half-empty bag of potato chips.
Yes, it's like if for dinner you found a half-empty bag of potato chips instead of eating prime rib. And so you're changing how nutritious that meal is because these diatoms are protecting themselves instead.
So going back to that transfer of energy, if you eat an apple, Randy's talking about like a 10% energy transfer. If you eat a whole apple, about 90% of the energy from that apple is wasted. Only about 10% goes to kind of nourish your body. And then with each step up the food chain, you lose a lot of that energy. So if we're talking about diatoms, and then we use, for example, something like a blue whale or a large filter feeding animal, they're feeding low on the food chain. If we compare that with, say, a great white shark, they are feeding from much higher on the food chain. So there are a lot more steps between the phytoplankton, the base of the food chain, and the great white shark than there are between phytoplankton and a blue whale. So it's a little more energy efficient for those big whales to feed low on the food chain. And I lost my train of thought. But you were mentioning that when you add another step, then you increase the inefficiency.
Yes, yeah, because that's right. Every time, if you have a 10% transfer, so if 10% of that diatom goes in that copepod, then only 10% of that diatom is going on to whatever else, right? So each time it's less and less of that primary production. So every step in the food chain means whoever's at the end is getting less and less each time you add something in there.
And you can kind of observe this in the, like if you took all of the phytoplankton out of the ocean and you dried it all out and you weighed it so that you're comparing biomass, and then you did this at different trophic levels. By the time you get up to the great white sharks or any kind of top predators, you have so many fewer of those top predators, not just in numbers, but also in biomass. Mm-hmm. So if you are just like just taking a bulk weight of all of these different trophic levels or these levels of the food chain. So if you are talking about sort of understanding how this, like the implications out in the ocean, out in the real world. So another question that I have is about the availability of the silica. So is that a limiting factor for them bulking up, or is there just an abundance of silica? Some plants will have different limiting factors like nitrogen or phosphorus or...
Right. That's a good question, and I think a lot of that is probably based on location. But as far as I'm aware, I don't think it's necessarily one of the major limiting factors. We can play with that and we can limit it for ourselves in the lab and see kind of what do they do when they can't get silica. But I'm not sure how much of a limiting factor it is in the environment.
And then if you're thinking about this in terms of the predators that they're exposed to, you are specifically looking at copepods, and so that would have perhaps you'd have kind of a different, much different implication out in the ocean if they are responding to dead diatoms versus copepods specifically, although copepods are very abundant in the oceans. But what about other predators or grazers?
Yeah,
you might call them grazers.
They have plenty of grazers, right? So that's also something that we would like to look at is this a predator-specific cue? Is this just copepods? Because a lot of the experiments done in the past have only looked at diatoms and copepods because they're such a famous, I don't want to say necessarily rivalry because it's not, it's a predation or grazing.
Like cows eating grass.
They have a relationship because they are constantly trying to out-evolve each other. Copepods have teeth made out of silica because it's easier for them to eat the diatoms with their silica test when they have teeth that are the same. So there's a close relationship between these two, and so that's why we focus on them so much. But I definitely think it's important to see, okay, is there the same response here with some other grazer, whether it's protozoans or crab larvae, or even just like bigger things like crabs, adult crabs. Like, is this cue present for any kind of predator, or is it just copepods? Or is it just diatoms dying? That's definitely something to pick apart, because in our lab, they're only dealing with one predator, but in the ocean, they're dealing with multitude.
So this is kind of, I like to describe, research projects sometimes as kind of like puzzle pieces. So a lot of times people, would like to hear how this is placed in a broader context. And so we, are placing that in a broader context. However, there are still a lot of unanswered questions.
Yeah.
And so your project is kind of addressing one little piece of that puzzle, and then there are many remaining questions to answer before you can really see that picture well.
Yeah, and a big part of that is their size. Working on such a small scale makes it difficult because your instrumentation is different. Like, you can put a couple of fish in an aquarium and it's no big deal, and you can feed them whatever and give them whatever cue. But, with working on such a small scale, I'm having to build things to figure out how do I cage a diatom that's, 20 microns. It's tiny, tiny things. And so that's one of the bigger challenges. But there are definitely still a lot of unknowns, which in science, you find that around every corner. But it is surprising to deal with that when it's such an important topic, because they are the base of the ocean's food chain. They're so important. It's a little bit scary how little we know.
Yeah. And then, so many questions come up. Like, would they respond the same to those organisms that are filtering them rather than crushing them?
Yep.
And just a lot of questions to sort of get a better idea of that picture of the way diatoms, their response in the ocean.
Yeah, and beyond that, I'm just looking at biological triggers, but what kind of ecological or environmental triggers exist? Like, does temperature and salinity and nutrients, silica, do those things also affect them bulking up, or any kind of other physiological response? There's a multitude of things that could be affecting any kind of response. Mm-hmm. So it can be a little daunting.
Yeah, well, that's why you take small bites and address one small piece of that puzzle at a time.
Exactly. I'm just trying to get a response so that from there I can hopefully address some of these questions and figure out, are there other predators that cause this cue? Is it the predator? Is it the diatom death? Like, what's going on? Tease that apart a little bit more.
Just with that kind of in mind, another question that comes to my mind is, if you think about this again, I think it's just kind of, we're more familiar and they're more visible, you know, these plant and animal relationships that we might draw, you know, analogies from. But for example, if you think about grazing on grass, you know, they're different. Grass is different. Grows on rhizomes. It has roots, which the diatoms don't. But then one thing that might be kind of analogous would be that the grazers, their waste would provide nutrients for their food source.
Yep.
So there's something understood about the way the diatoms grazers might sort of cycle that nutrient back to the population?
Yeah, I mean, fecal pellets can certainly fall and create more nutrients for the deep, which is already very low in that, right? It's hard to find food sources when you live in the deep ocean. And so, like, even dead sinking diatoms can provide that kind of waste to the deep ocean. You're looking at carbon export, which is also really important. So there's definitely, yeah, it is the same way as like cows, where those fecal pellets or any kind of leftovers, essentially, diatom particles, things like that.
Because it's only a 10% transfer.
Yeah, all of that can certainly be exported, or can, because they're so small, their sinking rates are very, very long, right? The smaller you are, the longer it takes for you to sink. And so they can also just kind of sink slowly and provide that kind of food or nutrients to any of those interstitial levels of the ocean.
So do you have kind of a big picture message that you'd like to leave folks with?
I mean, the biggest thing that I'm always trying to, I guess, harp on is that even though you can't see these guys, and even though they're little, I know they're not dolphins, but they are still important, and they're still fun to look at. Like, these pictures are beautiful, and people will create art from pictures of phytoplankton and pictures of diatoms. And so these beautiful little plants are a big part of the ocean's cycle, whether it's the food chain or nutrient and carbon export, all of that. These little tiny guys play the biggest role in that. So,
yeah. So just to bring that home a little bit, they're important to humans too.
Yes.
So even if you've never heard of phytoplankton, they're providing a significant percentage of the oxygen that you breathe.
Yeah.
And, you know, if you like to eat seafood, you know, you have phytoplankton to thank for it.
Yeah, if you like oysters are eating these. So there's that already. But yeah, oxygen is also, they're photosynthesizers. So they're creating a lot of the oxygen that we breathe, especially if you live on the coast and you're also eating that food.
So, I mean, I know it's maybe challenging and there's been some debate, but just to give people a ballpark, the percentage that has been landed on most recently that there's kind of a general consensus on is about 50% of the oxygen on the planet is produced by phytoplankton.
Yeah.
So it's significant.
I was going to say, I tell people it's every other breath you take, essentially, is provided by phytoplankton. Mm-hmm. So,
well, thanks for chatting with us.
Thanks for having me.
And thanks for joining us this morning, and we hope to see you for our next Boardwalk Talk.